унылая видно. Он что, болеет? Он ковидом болеет?
Нет.
Или так болеет?
Ну, просуда была, он сейчас сидит.
На самоизоляции?
Не, больничный собирает. Он уже в поликлинике.
Уже выздоровел?
Да.
Отлично.
Все
Все в порядке, звук включен, да? Good afternoon, guys. Yeah, I can see that you are collecting here. So, good afternoon, Rahul. Good afternoon, Rukaya, Rachavan, Kulot, Murti, and Donia, all other guys. So, and you see the today's topic. And today's topic is immunity against SARS-CoV-2. That is a newest coronavirus. Okay? I hope, as usually, you have a coffee in front of you, or tea, or soft drink, and you are going to listen what is going on there. So, and at the very first slide of our lecture, I would like to draw your attention at the very nice picture that I found somewhere in the internet. You see that the infection of the coronavirus comes through the lungs usually because there are a lot of receptors in lung tissues except for other parts of the body. And then as a result of infection, we have inflammatory signals that activate dendritic cells, monocytes, and macrophages as a major antigen-presenting cells. And as a result of this activation, so the release of a lot of different types of cytokines and interleukins may cause very strong pathological reaction that now is called cytokine storm. Can you see here all those molecules that are normally produced in any infectious disease? So they behave themselves really like, you know, I think this is like tornado, okay? So it means this is shown the dysregulation of the immune response. And the outcomes of this dysregulation are air respiratory distress syndrome, RDS, multi-organ dysfunction or multi-organ failure, hyperinflammation syndrome, that what we usually in the very scientific manner, that term we use to refer to as the cytokine storm, right? And the very pessimistic prognosis, it leads to death. My dears, the picture is nice, but the disease is not. And I would like to tell you that within December, I'm going to read 2 lectures on SARS and COVID together. So, the first lecture in immunology, and this will be the lecture about the immune response against SARS, immunopathology, in how the immune response will be developed, immunopathology within this immune response development, and immunotherapeutic drugs or immunotherapy and immunoprophylaxis. I would like to tell you from the very beginning that a lot of data are not really, you know, very stable because this is a very fresh new infection. There are so many different speculations and doubts about these things. So that's why what we see today is the nowadays understanding how it works. And by the way, we have a lot of gaps everywhere. We don't know why it happens so or not. So we try to explain it, but of course our explanations is quite far from ideal. I hope you should understand, you will understand this. Okay, guys? And next Thursday, I will read a lecture on the virology or about the SARS virus as itself, as a biological agent. Okay? So let's start with this, and today I think you will need all your knowledge in immunology. We have studied this subject for several months, September, October, November. So you know a lot about the regular normal immune response. So that's why I hope you will understand at least the basic ideas that we will discuss today. And of course, you also need to know what are the candidate of drugs and what kind of drugs are really licensed already. But mainly, of course, it will be changing very quickly. But mainly, you have to understand the approaches in which we develop the drugs against COVID. Okay? So now let's start. Just few words about the SARS-CoV-2 because we will have a separate lecture for it. And you can see that this is circular, middle size, uh, oval or coccal shape virus, right? So, and I hope you know already that it is called coronavirus because of the very specific structure and configuration on the surface glycoproteins that are called spike proteins. You can see them here, spike proteins. So as far as you see it under the microscope, you see like a halo around the virus. So this halo resembles the halo around the sun. So the so-called solar crown. So that's why these viruses get their name, coronaviruses. And the structure is not so complicated. So this is a positive sense, single-stranded RNA virus. What does it mean? It means that its RNA may be recognized as a messenger RNA. And it may go directly, as soon as the virus enters the body, it may go directly to the ribosome to start the protein synthesis. But what we know about this genome is that is the longest RNA virus genome out of all viruses, right? It takes something like 30 kilobases in length. And one more distinguishing feature of this genome, that this virus and the genome of the virus, we have 13 open reading frames. So that is unusual number for the viruses. So, and in the genome, you can see the that the translation occurs into polyproteins. It means that those polyproteins need to be cleaved by protease, usually cellular protease and viral protease. So then that was very few words about the genome. So we will discuss it. Oh, sorry. We will discuss it more accurate next week when we have virology. And just few words about the structure of the virus. So you can see the spike protein that are glycosylated trimeric proteins. And usually you know already that they bind the very specific angiotensin converting receptor. So this esterase on the host cell. And the particular conformation of the spike protein will make the binding site to the this angiotensin converting enzyme 2 on the host cell membrane. So, but then you're supposed to know that this interaction is not enough, and one more receptor, so supposed to participate in this because they have proteolytic activity, and it need to activate the spike protein to let it to bind with the AC2 receptor more closely and at the higher affinity. Then you can see here the membrane protein. And the membrane protein, except for it is the surface protein, it has very important function. It is an interferon antagonist. And we have quite a lot of copies on the surface of the virus. And this is an enveloped virus because these two types of proteins are incorporated into the membrane. Apart from this, there is a third surface protein that is called M protein. Envelope protein. So we believe that this viral protein may cause the cell lysis. So this virus is believed to be cytolytic. And inside the virion, we may see the nucleic acid, RNA, so covered with nucleocapsid proteins, with nucleoproteins, so N proteins. So they form the ribonucleocapsid together, right? So, and the nucleoprotein also have the activity as interferon antagonist. Why we are talking so much about interferons? So today we talk a lot about interferons. Why? Because you know that interferons have direct or indirect, non-specific antiviral functions, right? These are type 1 interferons, alpha and beta. And but it appears that it is good if we have not so much of those proteins. And it appears that if we have the overwhelming response that produces interferons, then it's going to be pathological. So, and as you see from here, the nucleocapsid symmetry is the spiral symmetry. So that's what we see on the picture. So we won't discuss some more viral properties. We go ahead. So I would like to tell you that the SARS-CoV-2 is not the first virus that appeared in the human population. So we won't mention all that may infect humans, but we have to mention at least two that appeared before the novel SARS-CoV-2 appeared. And they have absolutely different epidemiology. And I would like to underline that SARS-CoV, the first one, that appeared in 2002, infected only altogether 8,000 people. But the mortality rate was quite high, about 10%. So within the epidemic, it kills 774 people. And it was contained within 7 months and a half. It was hard, and the very serious efforts were needed to stop it. And the most difficult was to stop the spreading of the virus through international traveling through the airports. But then we managed to do it. So it was contained within 7 months, and then it disappeared from the human population. By now, it is not circulating in the humans anymore. And of course, that SARS virus was animal virus, and the infection was zoonotic, but very quickly virus learned how to spread through humans. Then the second virus, coronavirus, that caused the really serious health problems was MERS. Initially, it was called Saudi SARS because the effect that it causes, the very severe respiratory syndrome. So that was very much same in MERS. So, but then the epidemiology was absolutely different. MERS has never been stopped. So it is quite rare. It was quite rare infection. But since it arises in 2012, so it infects 2,000 and a half people and killing 35% of them. So see, the lethality is much higher than that of the, and by the way, by far we have, so far we have 866 deaths. So, but it is still circulating in the human population. And the natures of this virus are camels. And this virus had been isolated from camels and from humans, but I think this is more difficult for this virus to be transmitted from human to human. And finally, after several years, a novel SARS coronavirus appeared. And by now, and this is yesterday's figures, right? So by now it infects more than 63 million people. Can you imagine? 63 million people. And kills more than almost 1.5 million of them. And this virus is not going to stop. So, and we think that the peak will occur of the incidence and the mortality this winter. So today we'll be talking about this novel SARS coronavirus. So it causes an infection that is called COVID. So most people infected with the virus will experience mild to moderate respiratory illness and recover without requiring very special treatment. This is a majority of the infections. Although the older people, some children, and those who have some underlying serious medical problems like obesity or cardiovascular diseases or diabetes or chronic respiratory diseases and lung pathology, then those who have cancer, those people, and that's a lot of them, so they are more likely to develop serious illness. So it sounds very understandable, but that's just a part of the truth. Because sometimes we may have so severe diseases in young people without any underlying medical problems, without any bad habits. So sometimes we just cannot explain with all our knowledge about this virus right now why these or that patient develops so severe, so serious diseases. So we think that much more factors really that are listed here may influence the coronavirus infection in humans. So, and we see just the tip of the iceberg. You know, iceberg. So now, here we can see the schematic presentation of the immunopathogenesis of the SARS-CoV-2 infection called COVID. So when you can see here, but this is the general overview, right? Now step by step we discuss the steps that you see here in more details. So the SARS virus, through the angiotensin converting enzyme 2 receptor, binds and enter the cells. And by the way, so if we take a look at the expression of this receptor in human tissues, mainly they are expressed in small and at less extent large intestinal tract and in the lungs, while it may be expressed in some other parts of the body. But what we know is it is not expressed only in the brains. But then you will know, you might have heard that there are some neurological problems. That's maybe because everywhere we have blood vessels, and these receptors may be found in the blood vessels also. So then, the virus enters the cells, multiply, and of course, while it multiplies, the viral proteins will be produced, and the virus will cause the immune response development in the body. Of course, usually it infects macrophages, dendritic cells, and endothelial cells very often and at high proportion. Then, a result of this activity, so they start to be activated and produce cytokines and start to be working as antigen-presenting cells. So now you see that, so this is innate immunity stage. Then antigen-presenting cells were infected, so they recruit the T cells, they activate the T cells, and through formation of immunological synapse, the information will be transmitted about the antigen from the APC to the T cells. And then, of course, we may have activation of T cells and cytokine production and activation of TCD8 positive cells, TCD4 positive and TCD8 positive. And then, of course, as soon as the TCD4 positive cells are activated, except for cell-mediated immunity, it will cause the B cells activation. After the antigen recognition, then the B cells will develop plasma cells and the antibodies will be produced. M, then G and A, and the whole spectrum of antibodies. And by the way, we know already, and it had been checked, that memory cells will be produced by T cells and B cells. It is already known and confirmed by the data. But what we don't know here in this picture, because it exists just 1 year. Do you know that the first claim of new virus appearance comes on 31st of December? So, and the Chinese authorities declared that they found a new virus, 31st of December 2019. Just 1 year passed. So that's why it was not so much to follow the dynamic of the antibodies. So we know that antibodies are produced, but we don't know how long they stay in a body and how quickly they wane from the blood. That is a question mark. And saying we don't know for how long CD4 and CD8 positive cells stay activated, so but then the memory cells, T cells and B cells will anyway keep the information about the previous contact with the antigen. So the cellular immunity and humoral immunity will be developed. And as a result of it, when something will go wrong and the development of the immune response start to be uncontrolled, then we have in some patients the hyperinflammatory response that has the name the cytokine storm, and then hyperinflammation, And then as a result of all this, tissue damage. You see the picture? I hope you understand the picture. First, all the events occur, and then we have cytokine storm. And who to blame in this cytokine storm? Almost all are mentioned here, the most important. IL-6, 8, IL-1 beta, and interferon also 1 beta, IL-17, tumor necrosis factor, interferon gamma, interferon 1 alpha, and granulocyte monocyte colony stimulation factors. Now I think you can read all names over the cytokines and interleukins that are produced. So now that is the overview of the immunopathogenesis of SARS. Now we take a closer look at the particular stages of the disease. So this is a very first and very important stage. This is entry of the virus into the cell. So for the entry, two receptors are critical. So, and as we said, there's angiotensin converting enzyme 2. And one more, this is transmembrane protease receptor that should perform the proteolytic cleavage of S1. So the S protein usually binds with the AC receptor. Then a significant cleavage event takes place with the S spike protein. And by this proteolytic enzyme that we name already, so this proteolytic cleavage result in very tight adhesion to the protein, to the AC2 protein, and the fusion of the viral membrane with the host cell membrane. Then fusion occurs and RNA will enter the cell. So the entry of the RNA occurs by endocytosis. Okay, so after the release of the RNA from the endosome into the cytosol, viral genome can be translated into two structural proteins and polyprotein, which help in viral replication. And polyprotein need to be cleaved. Upon infection with SARS-CoV-2, genome host cell activates well-coordinated and rapid immune response. What happens then? So now, I would like to draw your attention to this. So, this is the, this is the predicted host immune response to SARS-CoV-2. And when the SARS virus appear in the nucleic acid of the SARS virus appears in this endosome, you're supposed to see here the so-called pattern recognizing receptors. So among pattern recognizing receptors, we may have TLR7, 8, then we also may have, and then you see this is a receptors located on the endosome but not on the surface over the cell. Then you can see that so some other receptors are here. Do you remember Rig-like receptors? And also we may see here mannose that are not found, not shown. Mannose binding soluble receptor, and they also C-reactive protein receptor is involved. So then several types of the receptors recognize mainly single-stranded RNA and so like here, but mainly like here, sorry, and double-stranded RNA. You know that double-stranded RNA is the most important triggering mechanisms to activate Rig-like receptors and node-like receptors, because this is recognized as pump. So see, this is single-stranded or double-stranded RNA are pumps for these receptors. Then we go ahead. So as a result of recognition, you can see that this recognition activates several signaling cascades. Do you remember what we call signaling cascades? When the one receptor activates some adapter molecules that transfer the activation from outside the cell or from the endosome towards the nucleus. So then the adapter molecules like D88 that you know already, or some antiviral mitochondrial MAVS, right? Mitochondrial signaling proteins. So then TRIF also may participate. This is not found here. So that depends on the interferon beta. So these adapter molecules will transfer the activation to the so-called nucleic factor, transcriptional factors. Why we call them transcriptional factors? I would like to remind you that these change the transcription of particular genes upon activation. So those transcriptional factors, the very important one is NF-kappa B factor, and also the factors that depends on the interferons activation. See, interferon regulatory factor. So this, for example. So, and then they help in nuclear transcription and translation. In the nuclei, these transcriptional factors bind with the DNA molecule and induce a production of the variety of different cytokines. First, if you are taking a look at the NF-kappa B nucleic factor, it causes the release by the cell of the factors that are called pro-inflammatory cytokines. And these are IL-6 and tumor necrosis factor C. This is inflammatory response. Then the transcriptional factors that depend on the interferon regulatory factor, like 3 or 7. So then they cause the development of very good at the beginning of the infection. Interferons alpha and interferon beta, they are called type 1 interferons. You know that interferon gamma is called type 2 interferon, and one more is found, then we will mention it, interferon lambda. But this is a very good interferon. So because they have direct antiviral activity. So thus the interaction that happened here in between virus and the cell, so fabricate an assorted set of first-line defense against the virus at the entry site. Okay, we go ahead. So take a look at the same picture, but the one more part of this picture. So as soon as the virus replicates in the cell, the virus also produces some viral proteins. Those viral proteins, through ion channel, can be uptaken into the cell. And by the way, the virus replication and the viral proteins leads to the development of the so-called inflammasome sensor. So this is the part in the cytosol that may result in the signaling pathways that activate the production of very strong inflammatory signal that is called IL-1B. So the signal IL-1B is the very important one. It may cause, first it is responsible for temperature generation in the body. So this is endogenous pyrogen. So you know this about interleukin-1, but this beta part is the most important for us. So, and then the overproduction of the interleukin-1 beta will cause so-called pyroptosis or pyroptosis of the cells. What does it mean? The cells start dying out of the exaggerated inflammatory response. So the SARS proteins and non-structural proteins NSP9 and NSP10 might also induce the interleukin 1 beta production through the hyperactivation of NF-kappa B factor. So can you see it here? So NF-kappa B factor also may upregulate the production of the precursor of the interleukin-1 beta, and then it will be released by the cell. So we can see very strong inflammatory response here. And finally, accumulation of type 1 interferons can activate the cell. See, it can cause the auto-activation of the cell that produces interferons. And again, the activation of the interferon regulatory genes that also will be able to produce the inflammatory cytokines, and these cytokines is pro-inflammatory chemokine, CXCL10. So, but this is pro-inflammatory cytokine. You may not know the name. So, and the same signal will improve phagocytosis if the phagocytes will be already infected with the SARS virus. So, in the activation of macrophages as well as NK cells will, but mainly the regarding cytokines, macrophage activation will cause the very intensive cytokine production, but also activate the antigen presenting function of the macrophages, and they will start activating cells. These are naive T cells or TCD4 positive cells by viral antigens presented together with MHC2. And then the T cells of both types, T cells and B cells, may may be activated, and they differentiate into CD4 positive cells, but that is a small mistake in the picture. The one naive cell cannot give rise to both CD4 and CD8, right? So CD4 will be produced from naive CD4 positive cells, and CD8 will be produced from CD8 positive cells. And then the immune response will be developed with the help of CD4 positive cells and cytokine release, and also participate in hyperinflammation syndrome. And then the activity of CD8 cells also will participate in clearing of the infection. So then, out of this picture we see that blocking of production of interferons or disorder in the signaling pathways of interferons may alter the expression and the antigen presentation function of macrophages, and may interfere with the effective antiviral immune response. So, at the very beginning of the infection, these signals are of utmost importance. Now you understand better this picture, so the presentation of the immunopathogenesis, right? So we discuss what is going on here, here, and there. And now, just summarize that the acquired immunity against SARS mainly depend on the Th1 cells. It is Th1 mediated immune response. So, but that is not surprisingly. We know already that Th1 immune response plays a predominant role in the adaptive immunity against almost all viral infections. So, and the T cell responses mainly depend on the presence of the APC, on the quality of the APCs, and on the cytokines produced by these IPCs. So, do you remember that we discussed that the quality of APC and the cytokines produced by this APC make so-called microenvironment for T cells and influence the differentiation of the T cells into Th1 cells and Th2 cells. And again, I would like you to remember that there is no chance that the only one type of immune response is developed in the body, either Th1 or Th2. It never occurs like this. So always both are developed by the balance between these two types of immune responses are different. We may have higher Th1 immune response, then the Th2 will be lower because gamma interferon produced by Th1 cells, they downregulate the Th2 cells and vice versa. If we have high Th2 immune response, then the interleukin 10 produced by Th2 cells, it downregulates the differentiation and proliferation of T cells into Th1 cells because it inhibits this. So that's why both immune responses will be developed. But of course, CD8 cytotoxic lymphocytes, they are capable of secreting very important substances that may kill virus-infected cells. And these are granzymes, perforins, and gamma interferons. And they are usually essential for eradication of the virus infection. And the CD4 helper cells, T helper cells facilitate the overall adaptive response, so they are important in B cell immunity. So that is, on the other hand, B cell mediated humoral immunity response is also very important. Why? It may have really protective role. Why? Because they producing of so-called neutralizing antibodies. The presence of these neutralizing antibodies also impacts the reinfection and prevent the reinfection. So, but then, within the acquired immunity development against COVID, an elevated level of chemokines and plasma cytokines, and please take a look at this. Interleukin 1, mainly beta. Interleukin 2, 6, 8, 10, 17. Macrophage or monocyte colony stimulation factor. Granulocyte monocyte colony stimulation factor. Interferons and tumor necrosis factor alpha, that's are major interleukins. They are associated with the disease severity. So, as a prognosis, the presence of lymphopenia and cytokine storm that occur in the severe cases may have a significant role in the pathogenesis of COVID-19. Persistence of cytokine storm might stimulate necrosis and apoptosis of the T cells and leads to their exhaustion. This cytokine storm is responsible for commencing of viral sepsis, followed by lung injury induced by inflammation, and other complications like very severe ear respiratory dysfunction syndrome, pneumonitis, respiratory failure, sepsis, shock, organ failure, and potential death. Severity of COVID-19 is associated with the decrease in the number of B cells, CD8 cells, CD4 cells, natural killer cells, and the decrease in the interferons production. Now, I like this picture very much. This is a picture that is called double-edged sword of the T cell response. So this is the sword of the T cells. So, and in early events, the T cell immune response clear the viral infection. So it is beneficial. But then in late events, the T cell response and the presence of the T cells exacerbate inflammation, and that is the early events, and that are late events. So, and cause maladaptive immune response that may result in very severe immunopathology. So you can see the diagram. So when we have the reduce of the peripheral blood T cell counts, the viral load, inflammation, and disease severity increase. My dears, that was found in the patients. So we don't know about every type, but then the very strong reverse correlation had been found between these two factors. The viral load, inflammation, and severity of the disease on one side, and the number of peripheral blood T cell counts on another side. And once they decrease here, the severity will increase dramatically. So this is a clue for therapy understanding. So therapies to boost T cell and downstream immune response may help clear virus it very early during the infection. That is the problem with the treatment of the COVID. So the treatment in the early stage and late stage are different. So, for example, if we use type 1 interferon for treatment to increase the number of the molecules at the very beginning of the disease, neutralizing antibodies and NK cell therapy, it will improve the state of the patient. What we have on another side of this double-edged sword. So, therapies, as soon as the cells start to be hyperactivated, they start by T cells. They start producing a lot of the receptors to bind with the molecules, but also a lot of cytokines. Therapies to inhibit T cell and downstream immune response may help to prevent the maladaptive immune response when it is used late during the infection. And examples include the immunoglobulins that may, again, the monoclonal antibodies against the leukocyte adhesive molecules that you see here. So the antibodies against, for example, granulocyte monocyte colony stimulation factor, the antibodies against interleukin 6. So it means we have to decrease the activity of these cytokines later in the disease. So we have two absolutely different approaches in treatment. So when within the clinical studies, 5 very important observations on T cells in COVID had been found. So first, lymphopenia is a hallmark of developed cases of COVID. and the degree of lymphopenia correlates with the severity of the disease. So at the early stages, the T cells prevent the viral clearance, right? Then, as a result, we may have T cell exhaustion. It occurs later during the disease, right here, right? So then the degree of the exhaustion correlates with the disease severity. So, I miss, what do we mean under T cells? You can see CD8 positive activated T lymphocytes, CD4 positive activated T helper cells, these are T cytotoxic, these are T helper cells. And by the way, T regulatory cells are also in these counts. Then the third one, upregulation of co-stimulatory molecules in APCs and T cells may propagate the maladaptive immune response. So we have to downregulate the expression of co-receptor, co-stimulatory molecules as, for example, CD24. Do you remember it should be expressed on the surface of the T cells? But we have to downregulate the expression of these co-stimulatory molecules that won't allow T cells to be further activated. Then number 4, increased frequency of pro-inflammatory T cells. So, and these are T cells that may produce the granulocyte monocyte colony stimulation factors, and those also T cells that may produce interleukin 6 and gamma interferon. And the fifth observation that we get from the patients is that the decreased frequency of T regulatory cells. Do you remember at the very first lecture in our department when we discussed the inflammation that occurs as a result of the microbe invading? So we mentioned that the interleukin 10 is a very important factor produced by T regulatory cells to downregulate T cell immune response. And Th2 cells also may do this. So lacking of this factor and decrease of the T regulatory cells also promote excessive inflammation in the body. So I hope you understand this. So from this we draw the summary from the of the immunopathology. So first, depletion and exhaustion of T lymphocytes. So impaired T cells, right? T cell depletion and exhaustion. Then lymphopenia is seen, and the T cell impaired, and so the dysfunction of the T cells occur. Then increase in neutrophils. While T cells decrease, the microbial infections increase, so the activation and the recruitment and neutrophils occurs, so the number of the pro-inflammatory cytokines will be increased. Regarding also the net formation, do you remember neutrophil extracellular traps? They also may account for the pro-inflammatory response. Then cytokine storm. When we have the activity of Th1 to activate monocytes that may produce granulocyte, monocyte, colony stimulation factors. So that would increase the release of interleukin 6 also, and the hyperactivation of the system to produce pro-inflammatory signals. Then the very dangerous signal is interleukin 1 beta that is produced, and elevation of this signal will account for cytokine storm development. And the increased activity of Th17 cells may lead to increase interleukin-17 development, that is pro-inflammatory signal, because Th17 activate monocytes, neutrophils, and macrophages to produce interleukin-1 and interleukin-6 that will increase the pro-inflammatory signal. So then antibody-dependent cytotoxicity, because the presence of antibodies may activate the antibody-dependent killing of the virus with the help of NK cells and macrophages. So these are mainly immunopathological mechanisms that occur in severe cases of COVID. So now, next slide. This is a potential approaches how the immunotherapy of COVID may occur. And by the way, you can see here more or less realistic approaches, but also we may have not very realistic approaches. So we won't discuss them right now. So this is not so realistic, is the cell therapy that is just in the stage of the development. So we will concentrate on these three: interferon-based immunotherapy, antibody-based immunotherapy, and cytokine storm management immunotherapy. My dears, today I won't touch any antiviral drugs. This is not immunotherapy. You know they exist. So in the next lecture in microbiology, we will discuss the antiviral drugs that can be used in the antiviral treatment. But this is a different one. This is immunotherapy. This is how can we manage with this disease using immunological preparation. So go ahead. First, and all together we will see three therapeutic approaches against COVID-19. So the first one is interferon-based immunotherapy. And from the very beginning, I would make a comment. So here we need to perform it in very early stage of the infection. And as we said about the interferons, this is important in the beginning, because these interferon alpha, beta, and even gamma, they have the direct antiviral activity. So as a source of interferon alpha, we use recombinant human interferon alpha 1 beta and recombinant interferon alpha 2 beta, 2b or 2 beta. So then interferon beta is also used, and it is used as a preparation that can be inhaled. It has a particular number, but then it is inhaled interferon beta 1. So then we have interferons, so-called lambda. My dear, this is a third type of interferons. With you, we studied, so you know the classification of interferons a bit different from what we studied before. So type 1 is interferon alpha and beta. Type 2 is interferon gamma. And type 3 is a group of antiviral cytokines. They consist of 4 interferon lambdas. 1, 2, 3, 4. Lambda 3 is also known as interleukin 29 and 28 A and B respectively, right? So, an interferon lambda 4. See, interferon lambda 1 is interleukin 29. Interferon 2 is interleukin 28A, and interferon lambda 3 is interleukin 28B, and one more interferon lambda 4. Their function is very similar to that of the interferon alpha and beta, first type interferons, but the intensity and severity of the process of their release mostly developed less problems. So they are not that strong like interferon alpha and beta. So they are used as a first line defense against viruses in epithelium maybe. So that's why some of them are tested for inhalation. So to protect the epithelium. So this is proposed interferon-based immunotherapy. Now antibody-based treatment. It means with the help of antibodies against virus, not against any other things or cytokines, but against viruses. This is called antibody-based treatments. So here the several preparations are tested, my dears. Mainly that what they test. So, and this is not in very common use yet. Of course, it is used for patients that need help, but they are not licensed everywhere yet. So, as a source of convalescence plasma, the hyperimmune plasma is used from those who survived and recovered from the infection. So, convalescent plasma, what does it do? Restricts viral infection and modulates the cytokine storm, which finally improves the respiratory function and outcome of COVID patients. Then the second preparation that is used against COVID is monoclonal antibody. Usually, nowadays it is monoclonal antibodies, human antibodies, that is generated as neutralizing antibodies from B cells of convalescent patients. What does it mean? We take B cells from the convalescent patients, perform hybridoma from them by fusion them with the B cell lymphomas, right? B cells taken from lymphomas. So then we prepare human hybrid cell. So these hybrid cells start growing, dividing, and producing monoclonal human antibodies, and we use antibodies that usually target the S1 subunit, spike protein subunit. So they are used as neutralizing antibodies. Of course, it in the process of the clinical trials, but I think this preparation can be really very expensive. Then intravenous immunoglobulin. See, this preparation is different from just convalescent plasma. So, because they are highly purified and can be given intravenously. Several mechanisms contribute to the anti-inflammatory, and they are shown in patients to cause anti-inflammatory response. Why? Anti-inflammatory effect. The inhibition, these antibodies may inhibit the Fc gamma receptor. So to not allow antibodies to bind with the Fc gamma receptor on the different types of the cells, for example, macrophages. Then suppressing the activation of lymphocytes and inhibit the function of the complement system, because there are anti-complement antibodies there in those who survive the infection. So this is the preparations for antibody-based treatment. And now finally we have the approach in therapeutic approach, that is management of the cytokine storm, and that is maybe of utmost importance, because these are most important cytokines, but there are some others. I just give you here an example, because we also may have antibodies that may block, for example, granulocyte monocyte stimulated colony stimulation factors, or antibodies that can block interleukin 17, also very important in the pro-inflammatory response. But then you may take a look at them later on. So now, blockade of the interleukin-6, one of the important interleukins. So two monoclonal antibodies are given for you as an example here. Tocilizumab, my dear, Zumab means that they are humanized antibodies. So we have the only very small fragment from mouse antibodies, antigen binding fragment, and the rest of antibodies of human origin. And sarilumab, if it ends with umab, it means there's a whole human monoclonal antibodies. They inhibit the inflammatory pathway induced by interleukin-6. Usually, they block the receptors for interleukin-6. Then interleukin-1 blockage, especially 1 beta, I would like to insist, beta. Anakinra, that is the recombinant interleukin-1 beta receptor antagonist. What does it mean? This preparation, when given, it blocks the receptor again. So, sanakinumab, that's the same. In several stages of the disease, that could be an effective approach to reduce the progression of the cytokine release syndrome and air respiratory distress syndrome. So it means they manage the mainly cytokine storm. So interferon gamma blockade, emapalumab, these are human monoclonal antibodies that usually inhibit progression. In patients, we see that it may inhibit the progression to the severe disorder of the lungs. So, and this is interferon gamma will be blocked. And then, of course, the one that you know already, these are immunosuppressive agents like corticosteroids, and we may have them developed for oral uptake or for intramuscular injections. Then they inhibit the expression of the pro-inflammatory transcriptional factors like NF-kappa B factor in the nucleus of inflammatory cells, but they are never used and is not recommended to be used in mild to moderate cases. They are supposed to be used in only severe cases. Okay, I think, guys, we finish with immunotherapy. That's what you need to know by now about the immunotherapy. And now we go for the immunoprophylaxis. So you had a lecture about the vaccines already, so it will be not so difficult for you to understand it. But before, I would like to discuss what generally the approaches that can be used to stop the virus. That is a problem right now. So basically, we may have three approaches to stop COVID disease. And we discuss the potency of these approaches. First, extraordinary restrictions on free movement and assembly, as well as aggressive testing to interrupt the transmission entirely. So that is the effectiveness of restriction measures. They interrupt transmission entirely, right? So because if social distancing, that's very important. So, for example, if we know we don't reduce the social distancing, social contacts. So then, if one person is infected, so it may infect 2.5 people, and after 30 days, in 5 days, after 30 days, these 2.5 persons may infect 400 something people. Okay. So if we perform the distancing, it is calculated that one person may mainly infect one person and a little bit. And this is the average, right? So then, after 30 days, the only 15 people will be infected. Can you see the difference? 400 in 30 days and 15 in 30 days. That's what social distancing is for. Then, all those that we see here, coronavirus prevention. So I hope you all know about this already. So, and for example, because I have some contact with the infected individual, now I am on self quarantine. So I'm wearing masks, sit at home, and don't communicate. So keep social distancing and don't communicate with anyone directly. That's what we may get from restriction measures. So please don't reject these measures. So now the second one. And that was at the very beginning of the pandemic. That was very popular thesis. Just wait until enough people get infected and develop so-called natural acquired active immunity that is called herd immunity. My dears, this is a nonsense. The herd immunity can be obtained by uh, just being sick or being infected. So, the term herd immunity was developed as an epidemiological concern that describes the state where a population is sufficiently immune to a disease that the infection will not spread within that groups. So, for example, if blue are not immunized and yellow are immunized and healthy, and the red one not immunized, sick and contagious. So, if no one is immune, all here, even couple of cases or people who are not immunized, sick, and contagious may spread through the whole population very quickly. If some of the population gets immunized by having infections, so this immunized by healthy, but still, so we have many susceptible individuals here. When the coverage with the immunity is very low, it doesn't matter so much. So, because the contacts are still very common. Contagious disease spread through some of the population still. The morbidity will be still very high. When it may work, I mean herd immunity, when it may work. It may work the only when the most population gets immunized or not susceptible to the disease. Even if we have some, so we won't see any sufficient increase in the spreading of the disease. Spread of the disease will be contained. But regarding the severity of the disease, can you imagine how many people should die out of those that are infected to develop the herd immunity? This is a nonsense. And even Sweden now that claim that they want naturally to get the herd immunity naturally. Even in that country now the very strong again restriction measures are developed to interrupt the transmission entirely. So that's why it is a nonsense to get the herd immunity naturally. So that means the people should be vaccinated. That's what we are waiting for. We hope as soon as we will be vaccinated, then we may contain the spreading of this disease. So the vaccination is the only way to develop the herd immunity here, right? So vaccination that could protect everyone for developing that is artificial acquired active immunity. right? So to develop herd immunity within a country. So now, what types of vaccines can be used? So I would like to present some. So the protein-based vaccines. This is a quite old type of vaccines. Then the two types that are very popular right now, viral vector vaccines. Both Gamaleya vaccine in the Russian Federation and AstraZeneca so-called Oxford vaccine, have you heard of it? They are of viral vector types. So then nucleic acid vaccine. So two vaccines made in the United States, they are mRNA nucleic acid based vaccines. So we'll discuss them a bit later on. So and then finally the virus vaccines. So killed or alive viruses that may or may not multiply in the body. Okay, so a few words about the important uh, uh, why vaccination is a bit tricky. Very important features of vaccine in general are safety and efficiency. Safety, you understand whether we have some side effects after this vaccine injection or not. And efficiency is when it is injected, whether antibodies or T cells against this antigen are produced or not. And one more is not given here, protectiveness, but that's very difficult to evaluate. So then let's take a look. So if we have the so-called replicating vaccines, these are live vaccines or divergent, do you remember, taken from the animals, or some vector vaccines, right? So they are very good and their safety is not, but their safety is not very high. See, safety increase from here to here. So it means that the dead antigens, not live viruses, but their dead antigens are much more safe because they cannot multiply in the body. But take a look at the efficiency. So if the higher vaccine is purified, so the lower the efficiency of the vaccine. So, and the least safe are most effective. Live vaccines are considered not to be not safe, but they are highly effective. So then, which vaccines development is most popular right now against COVID? These are vaccines against COVID. So the most popular is protein-based still. Many countries try to develop protein-based vaccines because these are very old, well understandable, very safe, and quite efficient. So, but then on the second place, we have viral vector vaccines, and the balance is different. Why? Because the viral vector vaccines would give much better immunity. Maybe they are a little bit not safe, less safe than the others. But then they give not only antibody immunity like by protein-based vaccines. They cannot cause T-cell immunity. But viruses, viral vectors may cause the very good T-cell immunity, cell-mediated immunity, and also antibody-mediated immunity. So that is a very good thing about these vaccines. Then nucleobased vaccines. These are newest. So they can be DNA vaccine or RNA vaccine. And you know now that American vaccines are both RNA vaccines. There are some advantages in this vaccination. Why? The lower or small doses needed to develop immunity, and they are quite cheap, so they will go to the ribosome, and the cell produces the viral protein itself. So, but my dears, by now, none of these vaccines, nucleic acid vaccines, is certified or licensed in Western world, and both in the world, right? So they are totally new. Of course, they are under study for about 10 years already. But then that is a very serious consideration that none of the vaccine that we use right now for any diseases are nucleic acid based vaccines. So many types had been produced as experimental vaccines against HIV, against Zika virus. Do you remember Zika in Brazil? So, but then none of them are licensed. That is the problem of these vaccines. So now let's take a look at the vaccines. The simplest one is protein-based vaccine. And out of those that work on it for COVID, 28 teams are working on vaccine with viral proteins. By the way, in China, there is a vaccine based on the protein, the spike protein of the COVID, of the SARS-2 virus. And also in the Russian Federation, Epivac vaccine is made up of the peptides that are analogs of the S-spike binding site. So, these vaccines are poor immunogenic. They might require adjuvant. And the adjuvant is the immunostimulated molecule of delivers together or alongside with the vaccinal antigen itself. And then it improves the immune response. So then the proteins can be incorporated into the virus-like particle. It is not virus. So do you remember we studied hepatitis B virus when the HBs antigen is incorporated in the just lipid membrane? That's something like this. There is no genome. This particle is not infectious. It is not really virus. We have just a membrane, lipid membrane with incorporated their viral coronavirus spike proteins. So they are not infectious at all. And several teams working on the virus-like particles out of those who work with the COVID vaccine. So they trigger quite strong immunity. But the disadvantage of it is very difficult to manufacture. But then the good thing is that it may cause the cell-mediated and humoral immune response. Then subunit vaccines. These are also the vaccine that we get a spike, the whole spike, or cover the linear epitopes recognized by T cells. That is also possible. So, and these are protein-based vaccines still. Okay, so live attenuated vaccines or weakened vaccines. A virus is conventionally weakened for a vaccine by being passed through the animal or human cells until it picks up mutations that make it less virulent. So, Codagenics in New York is working with the Serum Institute of India, a vaccine manufactured in Pune. So, to weaken coronavirus by altering its genetic code so the viral proteins are produced less efficiently. So I think there's no vaccine of this type that undergoes third stage of the trials in a lot of people. So they are in process. So, and I would like to tell a few words about the viral vector vaccines. They can be replicating and non-replicating. So, and that was in Russian Federation. That what they use in Russian Federation was newly approved Ebola vaccine as an example of viral vector vaccine that replicates within the cell. So such vaccine turned to be very safe and provoke strong immune response, humoral and cell-mediated immune response. Because while dendritic cells uptake these viruses, they will activate the CD4 T lymphocytes through MHC2 presentation and CD8 T cytotoxic lymphocytes through MHC1 presentation, sorry. So they cause very strong immune response. So the non-replicating, we won't discuss because they are rather used for the gene therapy, and the giant Johnson & Johnson, they are working on this type of vaccine approach. So a few words about the vaccines of this type. See, these are viral vector replicating vaccines. So the Gamaleya National Research Center for Epidemiology, Russian Federation, published the results already for first and second clinical trials, and this vaccine had been approved for human use in August without third stage clinical trials, and third stage is the large scale trials. So, but then it was really so urgent to develop vaccinations that that was certified in Russian Federation before they entered the third stage clinical trials. Now it is ongoing, using 40,000 population, and they are ongoing not only in Russia but also in Brazil, India, and I think somewhere else already. Okay, the second vaccine of this type, and Gamaleya right now will be used very soon within December for the vaccination of high-risk groups that are medical doctors and healthcare workers and teachers at school and some other high-risk groups. Then the so-called Oxford vaccine, this AstraZeneca, this is the one who will produce it. They developed, or the Gamaleya vaccine is developed on the two adenoviruses of serotype 26 and serotype 5. And it is given twice. The first dose on one adenovirus and the second dose in another adenovirus. Why it is good? What is the problem with these types of vaccines? So, you see here, the adenovirus, let's say this adenovirus, and this is a gene code for coronavirus spike gene. Okay? Code for coronavirus spike protein. So as soon as it is given to the cell, the body may develop the immune response against surface proteins of this vector. Then when it is given second time, the immune response may neutralize this viral vector and will not allow it to enter the cell. Do you understand what I mean? So it means the second type, when this gene with a spike is given for expression in the cell, so it should be given in another vector. So that is good about Gamaleya vaccine. So in AstraZeneca Oxford vaccine, they use the only one vector, Japanese adenovirus. That the configuration is same, so the gene for the spike protein is there. So when the virus-like particle enter the cell, the process of transcription and translation happens, and the proteins will be expressed by this antigen presented cell. Okay, right? So then, but the problem of it is that the only one vector, so that's why this vaccine also should be given twice. When it is given first time, it's okay, but then when it is given second time, the body may perform neutralization of this vector. So then the vaccination will be not so successful. That is the problem. So, and I think because of this problem, this vaccine is undergo now the additional studies, because there's some mess with the doses that they need to use. So then viral vector vaccines, and here I would like to DNA and RNA vaccines. I would like to tell this the last words about vaccines that are called Moderna and Pfizer and BioNTech. Moderna and the National Institute of Health of the United States jointly developed an mRNA-based vaccine consisting of the sequence-optimized mRNA encoding the spike glycoprotein encapsulated into lipid nanoparticle. So the dose escalating trial happens, and this vaccine induced both spike glycoprotein binding and virus neutralizing antibody responses in the recipients aged 18 to 55. And now this is the first vaccine that will be licensed, I think, and produced in the huge amount, around 40 million doses at the beginning. Then the second one, also Pfizer and BioNTech, have also developed a series, and this is the Pfizer, this is a German company, and BioNTech is the technological USA company. So this is a mRNA-based COVID-19 vaccine, again with lipid nanoparticles formulated, nucleoside-modified mRNA vaccine, elicited the receptor binding immunoglobulins determinant, receptor binding determinant immunoglobulins G and neutralizing antibodies. There are some side effects. So, but then these 4 vaccines right now that are presented, Pfizer and Moderna from the United States, finally, Gamaleya from the Russian Federation, and Oxford AstraZeneca from Great Britain. These 4 vaccines most probably, we hope, save the world. With this, I would like to finish my presentation. So, and we will have a class regarding the immunity against COVID. So, please read this, make the photos of the slide. So, be prepared to answer the questions. With this, my dears, I finish, and please, you are invited for the final lecture next week. That will be about virus itself, replicative cycle, origin of the virus, and so all this viral, for you it is already routine. Goodbye, my dears. Goodbye, Jasmine Sethi. Thank you very much that you stay at the, so listen to the end. Goodbye, my dears.